My name is Yonatan Perez. I'm a postdoc in Arnold Kriegstein's lab. And today I'm going to talk about cell type specific molecular changes conservation across AST subtypes. So I would like to start with a brief introduction. When I joined the lab in late 2018, I was fortunate to work with a senior postdoc in the lab at the time. And together we use single-cell genomics of idiopathic autism neocortices. And we uncovered that the expression of synaptic genes in the upper layers, cortical neurons, were preferentially affected. So
I'm sorry, Yonatan, we can't hear you really well. Maybe you can.
There's not much that I can do because it's
This is perfect if you can stay in this position.
Can you hear me now?
Try to be closer to the screen.
Yeah, I'll try to get closer to the microphone. Is it better?
It seems better. Yes, thank you.
All right. Sorry about that. So this has led me to ask key questions that I wanted to further explore in this follow-up study. And one of the questions was, what are the prenatal and postnatal cell type specific molecular changes in other forms of autism, such as syndromic forms? And the other question was, what's the degree of convergence or divergence between different molecular changes of idiopathic cases versus syndromic forms of autism? So this has led me to focus on duplication 15q syndrome, which is one of the leading genetic causes of autism, thought to account for up to 3% of all cases. It is characterized by hypotonia, motor delays, variable degrees of intellectual disability, epilepsy, and of course, autism spectrum disorder. It is caused by the presence of at least one extramaternally derived copy of a specific locus called Prader-Willi critical region, which lies on chromosome 15. And these duplications most commonly occur in two flavors, either as an interstitial duplication, which is shown in the bottom panel B, or as the most common form, isodicentric triplication, which is shown in panel C. On the bottom right side, you can see also a cardiotype from one of the patients. So in order to answer some of my questions, I adopted an experimental paradigm that uses cortical organoids to study molecular changes of duplication 15q prenatally, or to model prenatal changes of these using these organoids. Basically, I used iPSCs from dup15q donors and controls and a dorsal formalin differentiation protocol, and sampled cells for single-cell RNA sequencing at different time points that roughly corresponds to first, second, and early third trimester of cortical development. To explore postnatal changes of dup15q, I used single nuclei RNA sequencing of 49 snap frozen cortical samples from 11 dup15q donors and 17 neurotypical controls from 3 different brain regions that were previously shown to be associated with autism. And after generating the libraries and sequencing, I used Seurat tools to basically perform QC, normalize, and annotate, and perform unbiased clustering. So what you can see here on the top left is clusters, a UMAP plot that shows clusters from primary nuclei. So after QC, I was able to generate over 345,000 nuclei profiles. So each dot on this plot represents a single cell, and adjacent cells are sharing similar expression profiles and are likely to be clustered together. And each cluster is color-coded to represent a specific cell subtype. The panel on the right is showing some of the markers that we use to annotate these clusters. And using this approach, we were able to identify excitatory neurons from all the cortical layers, as well as subtypes of interneurons, some non-neuronal cells that were related to the vasculature, as well as all the glial cell subtypes. Using similar approach, we were able to gather over 106,000 single cell profiles from organoids. Again, using specific markers, we were able to annotate clusters, and we identified dividing progenitors, intermediate progenitors, radial glia, again, all the excitatory neurons from different cortical layers as well as astrocytes. After that, we used clustering techniques. So we color-coded the cells based on different parameters, for example, to explore the sample distribution across the UMAP space to make sure that each sample is contributing to most of the cell types, as well as by diagnosis to explore whether either genotype is either making new cells or missing cells, and also by region to make sure that each cortical region can contribute to the total compendium of cells, and also by days of in vitro development for organoids to make sure that the cells that the organoids produced make sense in terms of their chronological emergence as compared to in utero development. We then performed cell type specific differential gene expression. And for primary nuclei, we found over 7,900 differential expression profile in over 3,600 unique genes. For organoids, we found less genes that were differentially expressed, around 2,900 in over 1,600 unique genes. So these volcano plots on the bottom are showing some of the top differentially expressed genes in the excitatory lineage. And maybe one thing to stress out from this slide is that although the majority of gene dysregulation was not directly associated with the DNA driven by this DNA dose-dependent effect, some of the most recurrent overexpressed genes across all cell types and also in terms of magnitude were coming from the duplicated region, which is not very surprising. We then wanted to see what's the degree of convergence between differentially expressed genes in excitatory neurons coming from organoids and compare them to their corresponding cell type in primary tissue. And these Venn diagrams are showing differentially expressed genes for specific neuronal cell subtypes and its corresponding cell in primary tissue. And the p-value is hypergeometric p-value showing what's the chance for such an overlap to occur by chance. And to the right side of each of these Venn diagrams is a correlation, a Pearson correlation plot showing what's the degree of correlation of overlapping genes. So overall, despite I would say developmental differences and also technical differences between the two data sets, we can see that there's a high degree of gene overlap between organoids and corresponding cell types in primary tissue, which might suggest that some molecular changes occur early on during development and might persist in the adult brain. So we then wanted to ask, what are these genes that are differentially expressed in excitatory neurons? Which biological processes are these involved in? And we found that neuron projection associated terms as well as synaptic regulation associated terms were amongst terms that were both affected in primary and also organoid cells. We then wanted to explore what is the degree of convergence between duplication 15q to idiopathic autism. So in order to do that, we used the data set we generated in 2019 from idiopathic patients. And then initially we just compared clusters just to make sure that we can compare apples to apples. And this plot on the left side hand is showing just a Pearson correlation of these different clusters. And then we wanted to ask whether there's a specific cell type that is more vulnerable in these two forms of autism. And we performed gene burden analysis and found that, again, upper layers in both idiopathic autism and duplication 50q seem to be preferentially affected, which highlight this cell type to be likely associated with the autistic phenotype. So not only that this specific upper layers are preferentially affected in both of these forms of autism, they also share very high degree of gene expression overlap, and as could be seen by the Venn diagram on the top. And also they share a very high degree of Pearson correlation, meaning that the magnitude or the direct directionality of expression is very similar in these differentially expressed genes. And these are some violin plots that show a handful of these overlapping genes between the layers of idiopathic cases in duplication 15q. And maybe highlighting just a few of them, we found a lot of genes that are known autism risk genes, such as CNT and AP2, that was found to be overexpressed in both. So again, asking what are these genes, or what are the biological processes that—
Sorry, Yonatan, you have 1 minute.
Yeah, this is the last slide. Again, neuron projection associated terms and synaptic regulation seem to be the pathways that are preferentially affected in both. So to summarize this, I hope I showed you that organoids can be used to model some prenatal molecular changes of DUP15q. I've showed you that dysregulation of DUP15q was not driven directly by DNA dose-dependent expression, but yet duplicated genes seem to be amongst one of the highest and most recurrent overexpressed genes across all cell types. I showed that DUP15q molecular changes specifically in excitatory neurons persist in the adult brain, involving neuron projection and synaptic regulation. And the final slide, I showed you that upper layer projection neurons are preferentially affected both in duplication 15q and syndromic, which is a syndromic form of autism, as well as idiopathic autism. And they share substantial gene expression overlap and molecular pathway convergence. And with this, I'd like to thank you for listening and acknowledge all lab members who participate in this study, our collaborators, the tissue bank, which gives the tissues and funding, and finally to my supervisor, Arnold Priest. I would be happy to take any questions. Thank you.